Good afternoon, friends. In last lecture, I have discussed the composition of culture media and also physical requirements, then chemical requirements, then different nutritions which are required for growth of bacteria. Today we will see in next unit, that is second unit. In that first point, that is staining technique. So before that, what is staining? And what is the need of staining? Or what is stain and what is staining? Why is it necessary to stain the bacteria? We'll see the different staining techniques. So first we will see why we should be stain the bacteria. It means what is the need of staining of bacteria? The bacteria have nearly the same reproductive index as that of the water. Therefore, when they are observed under microscope, they are opaque or nearly invisible to the naked eyes. Different type of staining methods are used to make the cells and their internal structure more visible under the light microscope. Are you getting the need of staining? Yes, what I am saying. The bacteria are nearly having same refractive index as that of the water. When they are observed under microscope, they are unable to see by your naked eyes. Therefore, there is the need of staining of bacteria. There are different types of staining methods are used to make the cells and their internal structure more visible. Yes. Second thing, what is necessity? First, what I am saying first, that is for visibility, and second thing to see the internal structure more visible under the light microscope. This is the need of staining, or why we should be stain the bacteria. Second, staining technique help in observation of bacteria. For example, the microscope are of little use unless the specimen for viewing are prepared properly. Then microorganism must be fixed and stained to increase their visibility. Yes, second thing, microorganism must be stained or fixed in order to increase visibility. Then next, stain and staining. The bacteria are slightly negatively charged at pH 7.0. Then basic dye stain bacteria and acidic dye stain bacteria. Then simple stain, the aqueous or alcohol solution of single basic dyes. So we will see the what is stain. Stain is a substance that adheres to the cell and it gives color to the cell. Yes, what is stain? It is a substance that adheres to the cell and which gives color to the cell. Then the presence of color, it gives the cell significant contrast. So are this much more visible. Then the different stain have different affinities for different organism. Yes, keep in mind the different stain having different affinities for different organisms or different parts of microorganisms. They are used, it means the stains are used to differentiate the different types of organisms or to view the specific part of organism. Yes, what I am saying, what is stain? It is the substance that adhere to the cell and it gives color to the cell. Second, it in presence of color, it gives cell significant contrast and it is much more visible. Then the different stain having different affinities for different organisms or different part of organisms. Then they are used to differentiate the different types of organism or to view the specific part of microorganism. Then we will see the types of staining technique. In that, first simple staining and second differential staining. In simple staining, it means it is the staining in this technique there is use of only one or use of single stain, and in differential staining there is use of two contrasting stains separated by decolorizing agent. Yes, what is the difference between simple staining and differential staining? In simple staining, it is the technique of staining in which there is use of only single stain, and in differential staining it is also the technique of staining in which there is use of two contrasting stains and which are separated by decolorizing agent. In simple staining, the use of only single stain for visualization of morphological shape and arrangement of organisms, and in differential staining for their identification and visualization of structure. For identification, it is again subdivided into gram staining and acid fast staining, or gram stain or acid fast stain. And for visualization of structure, spore stain and capsule stain. Yes, for visualization of structure, pore staining and capsule staining. These are the different techniques or types of staining. First, simple staining and differential staining. So what is simple staining? It is the staining, or it is this technique of staining in which there is use of only single stain. For what? For visualization of morphological shape and arrangement. And second thing, that is differential staining, the use of different contrasting stain which are separated by decolorizing agent. There are two main purposes. First, that is identification, and second, visualization of structure. In identification, basically two techniques are used. Two staining techniques are used. First, gram stain, or it is called as gram staining, and second, acid fast stain, or it is called as acid fast staining. And for visualization of structure, there is use of spore stain, that is endospore stain or spore staining, and second, that is capsule stain or capsule staining. Next we will see one by one. First, that is simple staining. Yes, simple staining procedure and its mechanism. First, in simple staining, it is the method of staining in which, yes, simple staining is the method of staining in which the bacteria are stained by use of only single stain. The simple staining is also called as monochrome staining or positive staining. Yes, keep in mind, this simple staining is also called as monochrome staining or positive staining. For example, the examples of simple stain are methylene blue, then saffronine, malachite green, basic fuchsia, and crystal violet. These are the few examples of simple stain. Yes, what are these examples? Methylene blue, then saffronine, malachite green, basic fuchsia, and crystal violet. These are the few examples of simple stain. In simple staining procedure, cells are uniformly stained. Yes, in simple staining procedure, the cells are uniformly stained. Then we will see the procedure for simple staining. These staining techniques are also, we'll see in also practically. Yes, procedure for that. First, a clean grease free slide is taken. It means it should be sterile. A grease free slide is made by first washing the slide with detergent with excess of water, and then slide is passed through the flame. Yes, take a clean grease free slide. Grease free slide is taken, then this slide is washed with detergent with wiping the excess amount of water. Then the slide is passed through the flame. Then on this grease free slide, smear is made by using sterile nichrome wire loop and cell suspension. Yes, on this grease free slide, the smear is prepared. The smear is made by use of sterile wire loop. It means nichrome wire loop and cell suspension. Then this slide or this smear is allowed to air dry. Keep in mind, not artificial drying, it is natural drying. That is, this slide is allowed to air dry. It means natural drying. After the air drying, this slide is rapidly passed through the flame for 3 to 4 times for their heat fixation. It means in between or by the use of burner. Yes, after the air drying, this slide is rapidly passed through the flame for 3 to 4 times for what? Heat fixation. Then after the heat fixation, the slide is placed on the staining rack and flooded with particular stain. Yet after heat fixation, the slide is kept on or placed on staining rack and flooded with particular stain, and this stain is allowed to react for 3 to allowed to 2 to allowed to react for 2 to 3 minutes. After heat fixation, this slide is kept on what? The staining rack. Then it is flooded with it means add different or particular stain or specific stain, and this stain is allowed to react for 2 to 3 minutes. Then further slide is washed under running water. It means normal water. Then the slide is air dried and washed. Then the slide is air dried and washed, and this slide is observed under microscope. This is the simple procedure for simple staining. Yes, what I'm saying, what is the procedure? First, take clean grease free slides is taken. Then the slide is washed with detergent and with excess of water, and then slide is passed through the flame. Then second thing, on this slide, grease free slide, smear is prepared or smear is made by use of what? By use of nichrome wire loop or sterile wire loop and cell suspension. That is your sample, that is bacterial suspension. Then this slide is allowed to air dry. Then after air dry, as I said earlier, it is natural drying, not artificial drying. This slide is allowed to air dry. Then after the air drying, this slide is rapidly passed through the flame for 3 to 4 times for heat fixation. After the heat fixation, the slide is placed on the staining rack and add or flooded with particular or specific stain, and this stain is allowed to react. It means this stain is required 2 to 3 minutes for reaction. And further, the slide is washed under running water, and then the slide is air dried and washed and observed under microscope. Yes, this is the flow chart for procedure of simple staining. Take a clean grease free slide, then prepare a smear on slide, then air dry and heat fix the slide, then float the slide with specific or particular stain, then allow the stain to react for 2 to 3 minutes, then wash the slide under running water, then air dry the slides and observe the slide under oil immersion objective. Yes, this is the mechanism of simple staining. As stain has ability to bind a cellular components, these abilities are depend upon the charges which are present on cellular component and charges, and also the charges present on chromophore group of stain. Then bacteria has a large number of carboxyl group on its surface, and this carboxyl group having a negative charge. When this carboxyl group carried out ionization, the reaction it shows COO minus and H plus. That is carboxyl acid. What is formula for that? That is COOH is equal to COO minus plus H plus. The nature, this H plus are ion present In nature, these H plus ions are present on the cell surface and further replaced by other positively charged ion like Na plus or K plus, that is sodium or potassium. Now when these simple stains are used, it has chloride group, and further these stains are carried out dissociation. For example, if the stain is malachite green, it will carry out dissociation and it gives the free radical. This is the mechanism. What is the mechanism? That is the stain having ability to bind their cellular components, and these abilities are depend upon the charges which are present on cellular component, and also it is depend upon the charges which are present on chromophore group of stain. Then the bacteria having the large number of carboxyl group on its surface, and this carboxylic group having which charge, that is it carries the negative charge. When this carboxylic group carried out ionization, it shows the reaction, that is COO minus plus H plus. Then in nature, the H plus ions are also present on cell surface and further replaced by other positively charged ion, that is or like Na plus or K plus. Now when these simple stains are used, it has chloride group. Further, these stains are carried out dissociation. For example, if the stain is malachite green, it will carry out the dissociation and it gives the free radicals. As you know, the what is dissociation and what is association. That is MgCl is equal to what? Mg plus and Cl minus. And this Mg plus ion, it gives positive charge on chromophore group. When this stain is applied to a cell, this positively charged Mg plus ion replace the K plus or Na plus which are present on the cell surface. Yes, what I am saying, this stain is applied to the cell, then the positively charged Mg plus ions are replaced by K plus or Na plus which are present on the cell surface. Thus the ionic bond is formed charge in between positively charged Mg plus ions and cell surface. Yes, thus there is formation of what? Ionic bond in between the positively charged Mg plus ions and cell surface, and thus there is a result of staining of cell. This is the mechanism of staining of what? Simple staining. Then we will see the application. The simple staining procedure in the stain the bacteria, it is easy and it helps in observation under a microscope. Then second, it is useful in preliminary studies of morphological characters of cell, that is, yes, what is morphological characteristics? For example, size, shape and arrangement of cell. Yes, simple staining procedure, it's stain the it is easily and help in observation under microscope. And second thing, it is useful in preliminary studies of what? Preliminary studies of morphological characteristics of cell. What are the examples or what are the morphological characteristics? For example, its size, shape and its arrangement. Then yes, what is fixation? As I'm saying, heat fixation. Fixation which may itself consist of several steps. And what is the aim of this fixation? To preserve the shape of cell or tissue involved as much possible. Yes, what is the need of fixation or what is the role of fixation or what is the aim or what is the objective of fixation? To preserve the shape of the shape of cell or tissue which is involved as much as possible. Sometimes the heat fixation is used to kill or adhere and alter the specimen, so it will be accept the stain. Yes, these are some examples of simple stains, methylene blue, basic fission. It provides the color contrast but impart the same color to all organism in a smear. Yes, this is all about simple staining. Yes, simple staining, it is the simplest and actual staining process. It may involve the immerging the sample before or after fixation and mounting in dry solution and followed by rinsing and the observation. The stain can be poured drop by drop on the slide. Yes, it is also called as what? Fluided. This stain can be poured drop by drop on the slide. Yes, keep in mind this, the stain is added or it can be poured or it can be floated on the slide by drop manner or by drop or by drop wise manner. This is the simple staining. Then this is all about simple staining. We will see the next staining technique in next lecture. I will stop here. This is the all about simple staining. And the what is stain, then what is need of staining. It means after watching this video, you can able to explain what is staining, then what is stain, what is heat fixation, then what is the need of staining, and then different stainings, different types of or different techniques of staining, and in detail account of simple staining. Yes, I will stop here.